Hey everyone, welcome back to Biochemical. This is Gayatri. In today's session, we are going to solve some more questions from gene interaction. By solving the question, you will get a clear idea about gene interaction. So let's get started. So here is our first question. A hypothetical biochemical pathway for formation of eye color in insect is given below. Okay, let's see the pathway. Substrate X is colorless, is converted into colorless intermediate. Then this colorless intermediate is converted into brown pigment. But if there is presence of mutant allele A, small a, mutant allele A, then it cannot convert colorless intermediate into brown product. Similarly here, a substrate Y is present is colorless and the substrate Y, it cannot convert it into colorless intermediate if there is presence of mutant B. Okay, if mutant B is present, then this colorless Y, it cannot convert it into colorless intermediate. But if there is no mutant form of B, then this substrate Y will convert it into colorless intermediate, then it will convert it into orange pigment. So the two autosomal recessive mutants A and B are identified which block the pathway. What we have seen before, if there is presence of mutant A and B, then they will block the pathway. Considering the mutants are not linked, what will be the phenotype of F2 progeny if cross were made between the genotype? Let me write down the genotype. Small a, capital B with capital A, small b. And the F1 progeny are intercrossed. Okay. So it is asking about the phenotype of F2 generation. So let's cross. Then in F1 generation, what we will get? We will get capital A, capital B, small b in F1 generation. But here it is asking about what will be the phenotype of the F2 progeny. Okay, F2 progeny means what do you have to do? We have to self cross. Okay, so it is a type of dihybrid cross. So this is the phenotype for F2 progeny of dihybrid cross. Okay, and for the respective phenotype, I have written the genotype. For solving any question from gene interaction, what you need to remember is to remember that what is the phenotypic ratio for dihybrid cross at F2 of F2 progeny and their respective genotypes. Then only you can easily solve this question. Okay, let's solve this question. Here both the allele A and B are present in dominant form. Okay, there is no mutation. So what will happen? Then brown pigment will form, also the orange pigment will form, right? Because the mutant forms, they only block the pathway. Okay, as here the dominant forms are present, then of course the end result will be formed, that is orange and brown. Okay, let's see the next one. Here the A is present in dominant form, but B is mutated. Okay, the B is mutated. As the B is mutated, then what will happen? The pathway will be blocked and there will be no production of orange pigment, right? So here the orange pigment will not be produced, only the dominant form of A is present, that means the brown pigment will be produced. So only what we'll get here, we'll get only brown. Then go for the next. Here the allele A is mutated. If the allele A is mutated, then end product brown pigment it will not produced. But the B is in dominant form, that means orange pigment will produced. So it will result in orange pigment. Let me write down. And here both the allele A and B are mutant form. This is a mutant form. That means it will block both the pathway and there will be no production of orange brown eye colored type. So what will it result? It will result in colorless. There will be no colored product, it will be colorless. Okay, so now we have solved the question. So it's always better first understand the question, then solve the question, then only go for options. Otherwise you will get confused if you look at the option at first. Okay, so let's look at the option. We'll get 9 will be orange and brown, option C, and option, let's eliminate option C and option D. Okay, then go for the option A. 9 orange and brown. Yes, 9 orange and brown. 3 orange. Yes, 3 are orange. 3 brown. 3 brown. 1 colorless. Yes, 1 is colorless. So the correct answer is option A. Two plants with white flowers are crossed. White flowers are arised due to recessive mutation. Here it is mentioned that white flower, it is because of the recessive mutation. All F1 progeny have red flowers. Okay. When F1 plants are shelved, both red and white colored progeny were observed. In what ratio will be red flowered plant and white flowered plant occur? So here it is already mentioned that two plants with white flowers are crossed, and white flower is because of the recessive mutation. So it is a type of dihybrid cross, as I told you before. So write down the phenotypic ratio for dihybrid cross with their respective genotype. We have to find out the ratio of red flower plant and white flower plant. And the white flower plant, it is because of the recessive mutation. Okay, so where there is recessive mutation, there will be formation of white color flower. Simple. Here it is recessive mutation, B is mutated, so it will be white color flower. Here A is mutated, white color flower. Here both A and B are mutated, then of course there will be white color flower, because in question it is clearly mentioned that the white color flowers are arised due to recessive mutations. Okay, so here the 9th will be red color flower where the dominant forms of alleles are present. Simple. What will be the ratio? It is 9:7. 9 red and 7 white. Okay, so the correct answer is option C. It is 9:7. This is the simplest way to solve the question from gene interaction. If you will draw the Punnett square and do all the dihybrid cross, then of course that will definitely a time consuming, and in examination you don't have lot of time to do all these things. So simple thing, just understand the question, what it is asking about, then write down the dihybrid phenotypic ratio with their respective genotype and find out the what will be its phenotype, like the way I solved. Okay, a black Labrador homozygous for dominant allele is crossed with yellow Labrador homozygous for recessive allele. Okay, on intercrossing the F1, the F2 progeny was obtained in following ratio: 9 black, 3 brown, 4 yellow. So as we have discussed in my previous video, that the ratio for recessive epistasis is 9:3:4. So as we have discussed in our previous video about recessive epistasis, so this is the ratio of recessive epistasis, that is 9:3:4. Also I have a video where I have mentioned the trick to remember the different type of gene interaction ratios. Please go through that video. That will definitely a lifesaver video. Please do watch that video. So from here we conclude that by looking at this ratio, we conclude that yes, this is from the recessive epistasis. No more question. This is from recessive epistasis. So let's look at the option. So let's eliminate the option D. No, it is not complementary epistasis. It is also not dominant epistasis. So it will be either option A or option C. Look at the option A, recessive epistasis where allele E is epistatic to B. Yes, this one is correct. Option C, recessive epistasis where allele E is epistatic to capital E. These are the different, small e and capital E, these are the different form of same gene. So in epistasis, this is a type of non-allelic gene interaction. So this one is wrong. Okay, this is the allelic form. So the correct answer is option A. When two independent pure lines of P with white flowers are crossed, the F1 progeny has purple flowers. Okay, white color flowers are crossed, and in the F1 progeny has purple color flowers. So if you know about complementary gene interaction, where I have explained that by taking the example of sweet pea plant, by crossing the two white color flower plants in F1 generation, you will get purple color flower. So this is all about the complementary gene interaction. We conclude by reading this portion from question. The F2 progeny obtained from selfing shows both purple and white flower in a ratio 9:7. Okay. Here it is also mentioned that the ratio is 9:7. This question is nothing. This is about complementary gene interaction. And following conclusions were made. Okay, let's see the options. Two different genes are involved, mutation in which lead to the formation of white color flowers. Yes, two different genes are involved because it complementary gene interaction. It is a type of non-allelic gene interaction. Yes, two different genes are involved. This one is correct, and mutation lead to the formation of white flower. Q. These two genes show independent assortment. Of course, these genes show the independent assortment. This is an example of complementary gene action. Yes, this is an example of complementary gene action. This is an example of duplicate genes. No. So the correct answers are P, Q and R. Option D is the correct answer. Following biochemical pathway for purple pigment production in flowers of sweet pea. Colorless precursor, then allele A, it convert the colorless precursor 1 to colorless precursor 2. Then colorless precursor 2 is converted into purple pigment by allele B. Recessive mutation of either the gene A and B leads to the white flowers. So if A is mutated or B is mutated, then that will lead to the white color flower. Read the portion of question, then understand that, and what you understood, just write it down. A cross is made between two parents with genotype. Okay, that is given. Considering the two genes are not linked, the phenotype of expected progenies are. Okay, so if there is a recessive mutation, then that leads to the white color flower, which is given, and the genotype of parent is also already there in question. So let's do the cross. Capital A small a, capital B small b, and other parent is small a, small b, small b. That is the recessive parent. Test cross, right? So let's find out the gamete. This one is capital A, capital B. This is capital A, small b, small a, capital B, small a, small b. But here you will get only one type of gamete, that is small a, small b. So let's write down the progenies. Capital A, small a, capital B, small b. To this capital A, small b, small b. This one small a, capital B, small b. And these two small a, small b, small b. Okay, and in question it is mentioned that recessive mutation of either gene A or gene B that will form white color flower, right? So here look at both A and B are mutated, that means it will lead to white color flower. Here A is mutated, if either of them mutated, then that will be white color flower, so this one will be white. Here B is mutated, that means this one also lead to white color flower. But here both the A and B genes are in dominant form, so this one, it will lead to end product of biochemical pathway, that is the purple pigment, right? So it will lead to purple pigment. So what is the ratio? The ratio is 3:1, 3 white and 1 purple. Okay, so we have solved the question. Now we'll look at the option and find out the correct answer. Option B, 3 white, 1 purple. So thank you so much for watching this video. If you like this video, then please do share with your friends and don't forget to subscribe my channel.